All right, so what are we going to mainly concentrate on this thing? We're going to concentrate on your photosynthesis. How does photosynthesis happen? What are the factors that affect the rate of photosynthesis, right? So when we talk about photosynthesis, photosynthesis is the process by which plants manufacture their own food. Excuse me, alhamdulillah. Or instead of saying manufacture their own food, they make their own carbohydrates basically. From raw materials using energy from light. Now, when we talk about photosynthesis, so the equation for photosynthesis is that you have carbon dioxide and water. The carbon dioxide is absorbed by the plants, right? So the carbon dioxide absorbed by the plants in the presence of light will convert into glucose. Glucose is C6H12O6 plus O2. To balance the equation, we will need 6 carbon, 6 H2O, and 6 oxygen. Okay. Now, when we talk about light energy, so light energy is a requirement for photosynthesis, right? If light is not available, photosynthesis will not occur. That's the main thing. If light is not available, photosynthesis is basically not going to occur. Now, when we are talking about our photosynthesis, right? So let's talk about a little bit about photosynthesis. How does photosynthesis actually happen? Now, we are just going to go over a basic concept of it. We're just going to go over a basic concept of it. When you guys do your A levels, you'll study in a lot more detail exactly how photosynthesis happen, exactly how light energy helps in light energy actually helps in making their own food. You'll study that entire chemical reaction in detail, right? Now, but basically what generally is going to happen, let's go over the general process. The general process is this, that carbon dioxide is absorbed from the air. When we do the topic of transport and plants, I will go over the structure of leaf with you in detail. So you'll understand exactly how carbon dioxide moves in then. But carbon dioxide is absorbed from the air, okay? And water is absorbed from the soil. Right? And then this light, the light is trapped by a chloroplast. What is a chloroplast? A chloroplast is an organelle. What is an organelle? An organelle is any structure inside the cell. So a nucleus is an organelle. Cytoplasm is an organelle. Mitochondria is an organelle. So an organelle in plant cells that contains the pigment chlorophyll and traps the light energy. So the carbon dioxide will come from the surrounding air, the water will come from the soil, and the chloroplast, which have the pigment chlorophyll, will trap the light energy, right? And as a result, it will be able to break down the carbon dioxide and the water to make glucose. Now, once the glucose is ready, okay, what does the plant do with glucose? Once glucose is ready, once glucose is made, what exactly does the plant do with that particular glucose? So what does it actually do with glucose? Let's see. What we're going to see is that your carbon dioxide plus water is converted into glucose. Now, some of this glucose is converted into starch. Starch is the storage form of glucose in plants. Right? Starch is the storage form of glucose in plants. In your animals, the storage form of glucose is glycogen. Okay? In animals, the storage form of glucose is glycogen. In plants, the storage form of glucose is starch. Then it will convert it into cellulose. Cellulose is again, starch is a carbohydrate, cellulose is a carbohydrate. It's made up of glucose. And it is important for making the cell wall. Right? The cellulose makes the cell wall of the plant, giving it that support that it needs to actually withstand movement of water in and out and not burst. Then some of it will convert into oil. So you know how your seeds, right? Like you'll hear olive oil or canola oil or sunflower seed oil. So that sunflower oil, where is it coming from? It's coming from the seeds. Okay. So that's because the glucose is converted into oil. Then it can also be converted into sucrose. For the purposes of transport. Okay. For the purposes of transport, it can also be converted into sucrose. Now, the question that might come is that why sucrose? Why is it that for the purposes of transport, meaning that if I want the glucose to go from the leaves to the other areas of the plant, I need to actually convert it into sucrose first. Why do I need to convert it into sucrose? why don't I act? Why do I need to do that? Why can't the glucose just transport in the form of glucose? There are a few reasons for that. Okay. The the reason for that is because number one, glucose is reactive. Okay. Glucose is reactive, meaning that you can't transport it as it is without it taking part in some type of chemical reaction or without it disrupting some of the chemical reaction, right? Then when we are going to talk about our when we are going to talk about our glucose, so glucose is also soluble in water. Okay. So it has an osmotic effect. And the third reason is that, okay, these are the two basic reasons why glucose first has to be converted into sucrose for the purposes of transport, right? For transport to occur, glucose has to be converted into sucrose. Other than glucose, what other minerals are required by the plant? So a plant requires, along with glucose, a plant also requires nitrogen. Why does it need nitrogen? It needs nitrogen to make amino acids and proteins. Then it also needs some sulfur. Sulfur is also needed for the formation of proteins. Then it requires phosphorus. Phosphorus is needed for the formation of DNA. Then it requires magnesium. Magnesium is needed for the formation of chlorophyll. If any of these nutrients or any of these minerals are not available, the plant is definitely going to suffer. You guys should know what is the function of each nutrient and how the absence of that nutrient will affect your plant. So for that, I have a small table. Give me a second to share it with you guys. Hold on. All right. So, for example, if the plant has a deficiency of nitrate ions, right, they will have a poor stunted growth because obviously, if they don't not getting enough nitrate ions, they won't be able to make proteins, so they won't grow well. With phosphate, if they don't have phosphate, again you will have dwarf or stunted plants. Okay, and possible necrotic. Necrotic is dead spots on the leaves. With magnesium, again, you will get stunted growth and you will get yellowing of leaves. Why? Because magnesium is crucial for the formation of chlorophyll. Okay. If there is not enough magnesium, then you won't, the plants will be able to make chlorophyll. And calcium is also really important. Calcium also leads to stunted growth, but mainly you guys should know about nitrate, potassium, and magnesium. These three you guys should know about specifically. Now, when we are talking about our plants, right? So in our plants, like I told you guys, for photosynthesis, daytime is like light energy is necessary. So I'm going to consider three scenario with you guys. One scenario, let's consider that it's daytime. This is our plant. So during the day, because the plant will be undergoing photosynthesis, so the carbon dioxide will be absorbed by the plant and the plant will produce oxygen, right? Then what happens in darkness? In darkness, again, this is my plant. So in my plant, what is going to happen that it's not photosynthesis we will see. We will see the opposite of photosynthesis. What is the opposite of photosynthesis? Respiration. So the plant will be breaking down the glucose, okay? And as it will break down the glucose, it will absorb oxygen and it will release carbon dioxide. What happens when the light is dim? It's not daytime, but it's not quite nighttime. In this case, what is going to happen that, okay, in daytime, the in daytime, the absorption of carbon dioxide, right, is greater. In nighttime, the release of carbon dioxide is greater. Okay. But what happens in dim light? In dim light, you will see both, right? In dim light, you are going to see that your uh, rate of photosynthesis and rate of respiration will be equal to each other. So carbon dioxide is also moving in, but it's also moving out. Why? Because the light happens to be dim. So yes, the plant will be doing photosynthesis, but it will also at the same time be doing respiration because there is not enough light available. This point where there is a rate of respiration, rate of photosynthesis are equal to each other, we call this compensation point. Now, another important thing that you guys should know are the factors that affect the rate of photosynthesis. What factors will make the rate of photosynthesis increase and what factors will make the rate of photosynthesis decrease? The factors that affect your rate of photosynthesis are carbon dioxide concentration, number one. So, what are we going to write down about carbon dioxide concentration? Quite simply put, greater the carbon dioxide concentration, the faster the rate of photosynthesis. Just give me 1 second, hold on. Right. So greater the concentration, the faster the rate of photosynthesis is going to be. Now, when we are going to talk about the effect of carbon dioxide, so we will actually see a graph that looks like this. You guys should know how to draw these graphs, okay? On the y-axis, we have rate, and on the x-axis, we have the concentration of carbon dioxide. Your graph will look like this. As the carbon dioxide will increase, the rate of reaction will increase. And then you will reach a plateau, right? Now, let me introduce you to the concept of limiting factor. Why is it that as the concentration of carbon dioxide is increasing, one second. So it is a limiting factor. Limiting factor is that factor which is limiting the rate of reaction, meaning that carbon dioxide is the reason why the rate of reaction is lesser. If I will increase the amount of carbon dioxide, the reaction will become faster, right? So your concentration of carbon dioxide is the limiting factor. It is limiting the reagent. It is limiting the reaction right now. If you will increase it, the rate of reaction will also increase. But eventually you reach a point where carbon dioxide will have no more effect on the rate of reaction. Why? Because carbon dioxide is no longer the limiting reaction factor. Okay. Now, maybe it is some other factor. Maybe it is the amount of water. Maybe it is the sunlight, right? So it is no longer the limiting factor. Now, some other factor is the reason why the rate of reaction is not increasing. So one is carbon dioxide. Then we have light intensity. Greater the light intensity, the faster the rate of reaction. The graph will be the same. Okay, the graph you'll get will be the same. So the same graph you guys can draw for light intensity. The same graph you guys can draw for, one second. Light intensity is a factor. Let me just list down the factors first. Hold on. So light intensity is one of the more important factors. Your carbon dioxide concentration is a major one. The water concentration or the amount of water available is also a really major factor. Higher the concentration of your higher the concentration of the amount higher the amount of water, greater the amount of water, the greater your rate of photosynthesis will be. You guys can draw the exact same graph that I told you guys. You guys can talk about the surface area of the leaf. Now let's come on to another major one. Effect of temperature. Temperature will have a different effect. then all of the other ones. For all of the other ones, when you increase them, the rate of photosynthesis will also increase. And this will be the graph. The graph I have drawn for you, you guys can draw the same graph every single time, but not for temperature. Temperature will have a completely, or not a completely, but a different effect. Hajra, Al-Johara, can you guys tell me what the effect of temperature should be? We have studied something about this previously as well. I've taught you guys. So I want to see if you guys remember, or if you guys can link the two chapters together. We studied about it in another chapter. What should be the effect of temperature?
I think if there's a very high temperature, then the enzymes will get denatured. So it will be about the
Yes.
enzymes.
Yes. So your graph is going to actually look something like this, right? You guys have to relate this to the topic of enzymes. So initially, as the rate of reaction will increase, as the temperature will increase, the rate of reaction will increase, right? Until you reach your optimum temperature. So for optimum temperature is the temperature at which your plants are going to have the highest rate of photosynthesis, right? So, but as you cross the optimum temperature, the rate of photosynthesis will start to decrease. Why? Because the enzymes will get denatured. So this you guys have to relate to the topic of enzymes. As the temperature increases, rate increases. Since the molecules will gain kinetic energy, so there will be more collisions. So the rate of reaction will increase. Then, but as you go past the optimum temperature, once you exceed the optimum temperature, what will happen? The enzymes will get denatured. And as the enzymes will get denatured, the rate of reaction will decrease. So these are the factors that will be affecting your rate of photosynthesis. Now, there's another major thing that we have to talk about in another important factor that we have to talk about, and that is we have to go over a few experiments. Okay. So we have to go over a few experiments that you guys will find especially relevant to your paper 6. Okay. So for example, you guys should have an idea about how you can for example test for starch. That is a really important one. You guys should know about how to test for starch, or the effect of carbon dioxide on photosynthesis, or the effect of oxygen, or your effect of light intensity, right? So you guys should have a little bit of an idea about these experiments. So I'm going to go ahead and share some of these experiments with you. Just give me a minute. Hold on. The first experiment we're going over is the test for starch. You guys should know specifically the steps of this experiment and exactly how we're going to do it. Like, why is each step done? So you usually start off with boiling the leaf in water. Okay, that's step number one. You take your leaf and you place it in boiling water. Why? Because this is what the examiners will ask you, especially in paper 6. Why? Why do we need to boil it? The reason is to break down the cell wall. We have to break down the cell wall to be able to test for starch. Okay. Reason number 2, you will then place that leaf in ethanol and boil it again. Why do we boil it in ethanol? To remove the chlorophyll from the leaf. Why do we need to remove the chlorophyll? The chlorophyll gives it a green color. So the starch test gives us a color change. When we test for starch, we're looking for color change. So if the leaf already has a green color, observing the color change won't be easy. So we have to remove all the pigment from it. Now, so we place the leaf in the ethanol and the ethanol goes in a water bath. Again, the examiners can ask you, why is the ethanol placed in a water bath? Why can't we heat ethanol directly? That's because ethanol is highly flammable. It catches on fire really quickly. So we always heat it on water bath. Then after that, the leaf is washed in the water to soften it up. Okay. And then this is the main test for starch. Whenever the examiners ask you to test for starch, what are you going to say? Add iodine solution to it. Okay. If the starch is present, the leaf will turn black, blue black basically. Okay. The leaf will turn blue black. So this is the test for what? This is the test for your photosynthesis, for your starch, right? Now let's take a look at another test. उसको नहीं बैठना उसके साथ Now let's see the test for effect of light intensity. Okay, so over here you guys will see that we have added a solution, an indicator. We call it hydrogen carbonate indicator. The hydrogen carbonate indicator is an indicator for what? For the carbon dioxide concentration. Okay, so we are running the experiment in this way. The first experiment is a control. So the control we always need for comparison. Remember that. In every single experiment that you use, there always needs to be a control that you can compare with. So our control over here is simply a solution which has the hydrogen carbonator solution inside it, and we place in the light and it has no leaf. Okay. Now, when we talk about our when we talk about in the second test tube, we have simply placed a plant with the hydrogen carbonate indicator in normal sunlight. Okay. In the third one, we have completely surrounded with a sheet of black paper to represent complete darkness. And the fourth one, we are using a black paper, but not that dark. Okay. So it is dim light basically. Okay. Now, the hydrogen carbonate indicator works like this. If the carbon dioxide concentration is normal, it will be pink in color. That's what we will see in test tube number 1. In test tube number 1, it will be completely pink. Then if the carbon dioxide concentration increases, okay, So we will actually see that it turns from pink to yellow. So for example, in test tube 3 and in test tube 4, that's what we will see. We will see that it will turn yellow. Why? Because the plant is not performing photosynthesis, right? It is nighttime, it is completely dark, or it's dim light. So the actually in 3, not in 4. In 3, we will observe that it will turn yellow. Why? Because it is producing carbon dioxide, because the plant is doing respiration. In darkness, the plant always does respiration. Since it's doing respiration, so the amount of carbon dioxide will increase and the indicator will go from pink to yellow. In test tube number 4, we will see again pink color. Why? Because this is in dim light. In dim light, if you guys remember, we had studied that photosynthesis and respiration are equal to each other. So there will be no change. Then in test tube number 2, the carbon dioxide will decrease. Why? Because the plant is undergoing photosynthesis. Since the plant is undergoing photosynthesis, so it will break down the carbon dioxide to make its own food. So it will show you a change in color and it will become purple in color. So if the carbon dioxide decreases, it becomes purple. If it increases, it becomes yellow. And if it remains the same, it stays pink. That's how the hydrogen carbonate indicator works. Now, I do want to emphasize you guys, there are multiple ways of running the same experiment. Okay, so to check for light intensity, this is one experiment. You can also do another experiment where you can take a piece of paper, black paper, put it on top of the leaf. Okay, leave it in the sun for one day, come back the next day and test for starch. So the area of the leaf where you have placed the black paper, that area will have less starch. The rest of the plant will have more starch. So there are many ways of doing these experiments. Okay, I'm just going over one or two experiments with you guys. Making sense so far? Does anybody have any question? Any part that's not clear? Confusion regarding anything? Hajra, Al Johara, Mariam, everything all clear?
Yeah.
All right, perfect. Okay, guys, we'll go ahead and stop it over here for today and we'll continue in the next class. Before you guys go, hold on 1 second.